All right. Let's see. Got your life cycle quizzes. Yeah, overall they were pretty good. You'll see, I'll post a key outside my office and you can check it out. But you'll see check marks. And that's what I do is kind of go through life cycle and check the spots that I'm looking for, names, hosts, locations in some cases. And there should be a total of 10. There are a couple exceptions where I might do like half points for certain things. But that's what I use to kind of help me keep track of, the points and everything. I will say though that if you have like 10 checks and you like say something incorrect, you could still lose points. So on these life cycles, you have one where the eggs are consumed by the snails. So if you said that they hatched and the mirror city had penetrated the snails, well, obviously that's not correct. So you could have everything correct except that part, so you lose some points. But that's going to be the idea. You're going to have on the exam, you'll have a choice of 2 species. You'll pick one and you'll diagram the life cycles. All right, and we have 2 of those. And then on the final exam, I think we do 3. 3 of those for the life cycles. I am thinking that we'll probably have each multiple choice or multiple answer question worth 2 points. And that means then we're going to be close to 100 points on all of our exams for the total number of points, and the life cycles then would constitute about 20% of the total. So if each of them were only worth 1 point, you're looking at life cycles constituting 30 to 35%. And I kind of feel like that's a little too much, too heavy weight on those. But the life cycles are important, knowing our species and so forth. All right. Exams next week, Friday. Next week, Thursday and Friday, we have a job and a candidate here on campus interviewing. So it doesn't conflict with our class. There's going to be a teaching demonstration and there's a research talk. I'll tell you when those are, but if you can attend, that'd be great. That'd be great. So, any questions? Just no. All right. So we left off Diphyllobothrium. Not going to draw up the life cycle, but there are a few things that we need to talk about because we just ran out of time. All right. So get to here. Make sure I talk about it. All right, so eggs get released out into the environment. What happens then? This is just a quick review. What happens then? They're hatched, releasing what? Right, all right. And then what do the Coricidium do? Okay, yeah, so they're going to move up, negatively geotactic. Why do they do that? Yeah, so they're trying to get into the spot where that first intermediate host is. So what's the first intermediate host? Copepod. You know what a copepod looks like? Some of you, maybe? I think you talk about it in zoology. There we go. Good images. Here's zooplankton, right? Those are one type of copepod. Basically, it's the same type of copepod. I don't think anyone's talking about the parasitic copepods, but that's what a copepod is. Feeds on algae, bacteria, protozoans. So it's going to mistake the Coricidium for a food item and go ahead and feed on it. All right? So then what happens? Okay, so our Coricidium gets into the hemocoel, develops a procercoid, gets eaten by a fish. Right? Then what happens?
Either the fish infected is consumed by a fish in a mammal, so it's consumed by another fish. And
Oh.
that fish is consumed by mammals, or
Yeah, so you've got these two different routes, all to get back to the definitive host, which is a fish-eating mammal, right? Now, in our host, when the fish consumes that copepod, the procercoid's going to burrow out of the gut and get into the muscle, and that's a source of pathology for that fish. So if they get heavily infected, if they eat a lot of copepods, and a lot of those copepods have these procercoids, there's a potential to where that fish is going to be hindered. Their movement will be hindered so that they then get preyed upon more often than one that's not infected. All right? Now, the ability to go into these fish and just kind of repeatedly go into larger fish usually ends up in accumulation inside these quote-unquote paratenic hosts. Because a larger fish is going to consume smaller fish that have all been consuming these copepods. So let's say there's a 1% chance that a fish is going to consume a copepod. All right, so 1 out of 100 of these fish will have a copepod, and then a larger fish might eat 200 of these fish in the span of, let's say, 2 or 3 months as they grow in development. All right, that fish predator now has a chance of getting 2 of these instead of just that regular 1%. So you can get accumulation. All right, but question I have for you is, how does a parasite know when it's in a fish versus a fish-eating mammal? Temperature. Why the temperature?
Because mammals are endotherms.
Yeah, body temperature. So that's actually the trigger. So what triggers the maturation inside the host is body temperature. They get into the fish-eating mammal. It's going to be warmer temperatures in the gut. They get out, and instead of burrowing out of the gut into the muscle, it's saying, hey, temperature's right. Let's start developing to our adult. All right, to our adult. And then you have maturation. They release the eggs. Well, in Diphyllobothrium, they're going to dump out a lot of eggs, and they're actually going to do it over a relatively short period of time. Why? It's because of these guys. Where's my cursor? It's because of these guys. If we were to look at zooplankton populations, they change. The community changes throughout the year. All right. So there is a period of the year where these guys are very prevalent, very abundant. That seems to coincide with large amount of egg production and release by Diphyllobothrium in the fish-eating mammal. So a lot of things that kind of or a lot of adaptations that increase probability of transmission, and that's just one of them. All right. So we went over this. What about the pathology? All right. So our pathology is going to focus on, human or mammal pathology. Drop something. So most of the individuals that are infected are asymptomatic. And we're going to say humans can be a host, right? Some of us are fish-eating mammals. Others won't touch fish. They won't touch fish. So most infections, asymptomatic. You'll have the tapeworm. You don't even know you have it. But you could exhibit some increased fatigue or just general weakness. You have your immune systems fighting this worm in your gut. All right. The parasite may cause some irritation of the intestinal lining, but it doesn't have hooks. It's not feeding on epithelial tissue or gut epithelium. All right, so it's not really causing a whole lot of damage. Maybe inflammation, immune system. So, you could exhibit fatigue and weakness. 1 worm probably isn't going to do that. 2 worms probably isn't going to do that. But if you had maybe 10 or 15, perhaps then you start seeing some of this fatigue and weakness. The important thing, one of the key features of infection by Diphyllobothrium is something called pernicious anemia. All right? So just anemia in general. What is it? What do we know about anemia? What do you know about anemia?
Low iron.
Not quite. What's that? Decreased ability to carry oxygen. Why? Low red blood cell counts. So anemia is really kind of characterized by low red blood cells. Question is, how do we get there? Zach said iron. So iron deficiency anemia is one of those ways that you can approach it. Another way is this, pernicious anemia. So now you have a decreased size and hemoglobin content, not necessarily red blood cells, but decreased size and hemoglobin content of red blood cells, if there are RBCs, due to a reduced ability of the body to absorb vitamin B12. Our body needs B12 to create that hemoglobin. All right, so hemoglobin is what locks up your oxygen, right? Dumps it out in our capillary beds, right? Tied to partial pressures of oxygen and so forth. That should be physiology. Maybe you learned that intro to bio. I think I was introduced to that freshman year before. I don't know when you guys might have learned it. But yeah, when you talk about body's ability of carrying oxygen, yeah, that's low red blood cells, decreased size, hemoglobin contents, you can't carry as much and so forth. Now the question is why? Why does this happen with infection by Diphyllobothrium? This part right here. So B12 is needed for hemoglobin production. The worm itself will absorb 80 to 100% of all dietary B12. So the worm gets it before you get it. And that's a problem. That's a problem. Now, what's kind of a bit interesting is that almost all of the cases are in Finnish people. So people from Finland or their descendants. So this may actually be something tied to some genetics component. I mean, you can go to ethnicities and so forth, and you've got, you could say clades, clusters, genetic clusters.
Is that the pernicious anemia that just happen, or cases that happen related to this tapeworm?
No, just related to this tapeworm. Just related to this tapeworm. Yeah, just related to this tapeworm. So, yeah, and it's one of these things. One of these interesting things. You could say, well, it's all dietary. No, you take supplements, these worms will suck it up for some reason. Why they use it? Good question. You can look it up. I don't know if anyone's figured out why they do that, but they do. So pernicious anemia, if you exhibit this, good chance you might have one of these tapeworms. But other key point here is that not all anemia is due to iron deficiency. All right, we will see other cases of anemia pop up. So, any questions? Other thing that we could get with this parasite is something called Sparginosis. Now, this isn't just for Diphyllobothrium. All right, Sparginosis is a generic disease caused by infection with pleurocercoids. All right, so any tapeworm that has this procercoid to pleurocercoid or like pleurocercoid 1, pleurocercoid 2, any of these tapeworms that have pleurocercoids have the potential to produce sparginosis in a human. All right, and that's why I talk about it here because Diphyllobothrium has it, but we really can't identify these worms, like what type of parasite you have, until we do some sequencing, or we take them out and we infect another host and get the adult worm out of it. All right, most likely cases are Diphyllobothrium, and this would be a case where you happen to, where the parasite just doesn't know it's in the right host. And key one would be here, and I list possible routes of infection. You accidentally consume a copepod. So you go out hiking, you look at a mountain stream, you say, oh man, this water's great. It's rolling over the rocks with mosses, and you say, this is super clean. You take your canteen or your water bottle, you soak up, and you're drinking copepods with all these procercoids in there. Possibility that one of them penetrates out of the gut into the muscle and develops this sparginosis. So here's like one. It's actually in the eyeball. That's it. In the eyeball. Second, perhaps you would eat an insufficiently cooked amphibian, reptile, bird, or mammal. So we know that in Chinese culture, there are some that would eat raw snakes, and that seems to be a source of infection of the Sparginosis. I say this third one is probably most mind-boggling, but I hate to put down witch doctors, but witch doctors might prescribe something like this. Say you have a cut, you have an ulcer develop, and then they come up with this idea of, hey, we're going to take muscle and viscera and grind it up and then apply it as a poultice and say, this is going to heal it up because you got all the good stuff that's in there. All that good stuff could be pleurocercoids that then migrate out of that tissue and into yours. So that's a possible route. Again, you're not consuming it, but it was in the meat, in the tissue that you put on your own open wound, and they migrate into you. So 3 potential routes. Is one more common than the other? I would like to think that it's these 2 are the most common routes. If this is the most common, we need to do better educating about medicine, educating people about the benefits of modern medicine. What type of pathology? Usually it's just these subdermal lumps. So you'll notice a lump on the skin. Might not notice, pain. Maybe there's some pain as it was moving initially, but once it stops, it's going to usually just stay there and just hang out. Proliferation via splitting is rare, so it's when we talk about asexual reproduction in some of our other tapeworms, some of them will reproduce, but it's pretty rare for those species. All right. Questions? All right. Next order, Cycloflidia. So that was Pseudophyllidea. The dominant Scolex type was Bothria. In our order Cyclophyllidea, the dominant Scolex type is acetabular. So a sucker with Scolex and usually has 4 suckers. And they're going to look like the suckers of the digenetic trematodes. In these proglottids, the parenchyma is divided. We'll have a distinct medullary region, so inner, and then an extensive cortical region, which was more of the outer type of region. We're not going to really be able to see those or distinguish between those 2 regions, but we will be able to see this next one. Members in this order have a single compact post-ovarian vitelline gland. Instead of having follicular vitellaria, like we've seen in the digenetic trematodes and in Diphyllobothrium, now our vitelline gland is compact and compressed. And it's going to look like another just darker staining organ. Right, you see it in Taenia, you see it in, you can't really see it in Echinococcus because our specimens are overstained. We can see it in Dipylidium. Right? It's going to be post-ovarian, so it's posterior to the ovary. These guys usually have a rostellum that is armed, meaning a rostellum that has hooks on it. All right. And then our genital pores are usually lateral, one side or the other, or alternating sides. There is one family, though, that has median genital pores, but we're not really going to talk about it, so it's more of like FYI. The tapeworms of birds and mammals, most of the tapeworms of birds and mammals will fall in this order. We've had examples of it. So this is kind of just a classical proglotid for Cycloflidians. Right here we have our bilobed ovary. All right. We do have several species that we're going to talk about here. We're going to start in the family Taenidae. All right, so this contains the largest tapeworms in this order. It also contains tapeworms that are the most medically important. So yeah, you could say Diphyllobothrium causes pernicious anemia, which isn't really good, and that's true, but we can treat it. We can give it antihelminthics and clear it. There's some significant significant pathology associated with Taenids. We're going to be talking about that. In this family, the ovary is bilobed. We have Taenia examples. This is Taenia saginata. So we've got our bilobed ovaries, we have our vitellarium, we have our genital atrium at both male and female. You're going to be able to see that vast difference. I think a lot of you were asking about it. Right? That's when you see it, there's this squiggly part. That's the vast difference. And serous sac and serous. We did have some difficulty with the vagina, and that's because sometimes the vagina ends up being behind vas deferens in our view. You can't really see it. But others, you'll see it as kind of like a line. I'd say almost like a hair. It might look like a hair in the specimen. This is just generic Taenia, just to kind of clarify some structures. The metacestodes in this group are various types of bladder worms. All right. And what's kind of interesting is that mammals are going to serve as the intermediate host. Ready? So we're kind of getting to terrestrial life cycles at this point. All right, so we're going to start with Taenia. We have two species that we're going to talk about. Fortunately, the life cycle is similar for both. All right, we have the beef tapeworm, which is Taenia saginata, and we have Taenia solium, which is the pork tapeworm. All right, so what we're going to do, we're going to go ahead and do the life cycle. There's my cursor. We'll put this slide back up. Let's see if you can get the rest of it. I'm going to say this is a fairly simple life cycle. I forgot my chalk. I had to deal with tiny pieces. All right, Taenia solium, just generic. Humans are going to be our definitive host. We're starting to see, though, that maybe Taenia can go into some other carnivores. That humans are our primary host, where the adults are in the small intestine. Adults are in the small intestine. All right, our eggs are going to get out in the feces, but egg release will be apolysis, which means what? What does that mean, apolysis? Take a guess. Consult with each other. What do you think? What's that? What is? They're breaking out of what body?
Is this uptake by into the small intestine?
So this is where our proglotid detaches. Apolysis is when our proglotid detaches. So this will be a gravid proglotid, detaches, passes with the feces.
then it's proglotid, right? Proglotid has general structures, but these have already degenerated because it's just sac full of, it's just basically a uterus full of eggs, right? But it also has muscles. So our proglotid is going to come out in the feces and then wiggle around. And really, probably wiggle around and try to get their way out from deep within the pile of feces. All right? Because they need to get out where they then will dry out and rupture. And that now will allow the eggs to be exposed. This is where people would think that they see like cucumber seeds or potentially melon seeds in their feces, and these things are moving. So that's kind of how you can, cucumber seeds don't move, proglotids do. All right, so they dry out and rupture, eggs are out, they're on the feces at this point, and then the eggs are going to be spread by flies because flies get attracted to feces. They're going to be spread by flies. Eggs are going to end up on some food items that our first intermediate host then consumes. All right, so I'm just going to put intermediate host because our intermediate host will vary by species of Taenia. Once they consume it, you will have the oncosphere that is in the gut. So the egg hatches, releasing the oncosphere, and then that will burrow out of the intestine, get into various tissues, muscles and tissues, and develop into a cysticercus. Cysticercus larval. So fat and muscle tissue. Intermediate host, cattle or pigs, depends on the species that we're talking about. And then these cysticercus larvae, depending on the species of Taenia, can replicate in the host. And then once they're at cysticercus stage, we can then become infected by consuming muscle and fat of this intermediate host. All right? Now, cooking will kill it. So we're talking about undercooked meat. Once we get in the human intestine, bile salts serve as a trigger. They trigger evagination of the scolex. So the cysticercus has basically, this bladder type thing where the scolex isn't completely invaginated. It's inside out, inside that surface. They detect the bile salts in our small intestine that says, ooh, I must be in the right host. The scolex everts, all right, and then attaches, and now we start getting development. And it takes anywhere from 2 to 12 weeks to start shedding proglotids. 2 to 12 weeks. Questions?
His life cycle is for both types of Taenia, right?
Yep, so we have Taenia saginata and Taenia solium.
The beef tapeworm is Taenia saginata because the intermediate host is cattle. Pork tapeworm, Taenia solium, is so named because its intermediate host is pig.
You say the flies spread, the eggs just stick to the fly?
They do. They stick to the fly and they fly around. So when you're out having a picnic and black flies are around, just think you could be ingesting parasite eggs. Oh. Yeah, that's great. All right, so what's
So
that?
when you ingest them, is it guaranteed that they're gonna, I guess, live in your body, or is it just a chance that when you ingest them that they might go on into like adulthood?
The eggs will produce the larval stage,
Okay.
and that is where our pathology comes into play. So we'll talk about that. All right, so Taenia saginata, the beef tapeworm, is the most common human tapeworm. It's the most common human tapeworm. We get it by eating undercooked meat. Think when you choose to eat a raw steak, there's a potential for that. Beef tartare. That's raw steak, raw meat. And I'll put a catch to that. Our food supply is actually very safe. Animals undergo testing. When it goes to the slaughterhouse, they take samples. They test for antigens of the parasite. They test for antibodies. I mean, they do testing. If this pops up, I don't think they're gonna actually put it into the food supply. So our food supply is pretty good. But if you buy backyard stuff, you go to your buddy's house. He's like, "Yeah, we're gonna butcher this and we're gonna cook it over the pit." Yeah, they're not doing that same sort of testing. Right? So with difference between these two, the beef tapeworm, so saginata, lacks a rostellum. Solium does. It has a rostellum, and we can see it. And this is an armed rostellum, and it probably goes out and helps it stay attached to the intestine. All right. These guys, saginata, is 3 to 5 meters in length. That's the adult. The adult could be up to 20 meters. So we kind of mapped out this room. Tapeworm down to that wall and back in your intestine. That's a big tapeworm. Right. And we'll talk about pathology when we do have the human tapeworm. Taenia solium is presented. We can be a host for the adult, but this is probably the most dangerous hookworm. Not because of the adult stage, but because of the larval stage. If we ingest the eggs, we can now serve as an intermediate host. So those cysticercosis larvae go out to the fat and body tissue, fat and muscles, and other various body parts, where they will insist and cause pathology, and we'll talk about it. These guys are a little bit taller, or a little bit smaller, up to about 10 meters, with an average of 2 to 3 meters, so they're going to be a little bit smaller in size. Here. All right, so the pathology that we see depends on the stage of the parasite. If we're infected by the adult, it's usually called teniasis. Not tinnitus, that's like ringing of the ears. This is teniasis. The pathology associated with this is similar to the pathology of other large adult tapeworms. So if we get an adult tapeworm, usually it's asymptomatic, but we could have moderate symptoms, minor to moderate symptoms, which could include dizziness, abdominal pain, some diarrhea, nausea, intestinal obstruction, only if we have a lot of tapeworms in there all at once, and potentially loss of appetite. So again, just some sort of GI discomfort. Also get feeling of like fatigue and weakness, just like that Philibothriem, but this is just general symptoms of an adult tapeworm infection. With Taenia, though, they don't absorb a whole lot of B12, so we don't see that pernicious anemia tied to it. That only seems to be associated with Diphilobothrym. But the other aspect is you may have some psychological symptoms due to like the wiggling of the proglottids. So just you see it, and then it just kind of makes you start imagining things, right? If I say, Andrea, I'm going to use you. If I tell Andrea, hey, do this, And she's like, why? If I told her, I think you have a spider crawling on you, right? And it's not, that's going to put it in her mind that for the next 5 or 10 minutes, she's going to be constantly being aware that maybe I have something crawling on me, right? And start imagining some things. That's kind of psychological effects. And that psychological effects could apply to all parasites. So if you sense you might have a parasite, then you start imagining things. I would say imagining things. Some of them are real. All right, some of the nauseas weren't moving around, just kind of causing, you could feel it. All right, so the adult stage, pretty minor. You can get it treated. If you see these moving proglottids, check out a doctor. Pretty simple to take care of. Ready? The real concern is if we ingest the eggs. And generically, that is called cysticercosis. This is infection with cysticerci, or larvae of the type Cysticercus. All right, we only get this from T. solium, from Taenia solium. You don't see that with Saginata. You don't see that with Saginata. The symptoms and the severity of cysticercosis really depends on the location of their cysticerci. All right, so if these eggs hatch and the oncosphere penetrates out of the gut and goes to skeletal muscle, the skin, the liver, fat tissue, and then develops the cysticercus, normally it's going to be minimal pathology. You can get some pathology if you have a very heavy infection, like if you load up on eggs, then yeah, you could get pretty significant, but normally it's minimal pathology. If that oncosphere goes to the eye and develops into the cysticercus, you have what would be called ocular cysticercosis. So the cysticercus in the eye. And it could lead to blindness, ultimately. Could lead to blindness. Why? It's because sometimes during the penetration of the oncosphere or the developing cysticercus is going to become larger, right, from where the size of the oncosphere. You're going to cause pressure on various tissue and cause necrosis. So that necrosis in the eye would cause some irreparable damage to the retina, the iris, or the choroid, which is the vascular area between the retina and the sclera, and we don't repair it. Our body wouldn't repair that. And if you get that, then you have potential for blindness. Maybe it starts out as a blind spot, or it could progress to full-on blindness in that eye. The other possibility is that the oncosphere travels to the brain, in which case it's called neurocysticercosis. Unfortunately, neurocysticercosis is rarely diagnosed until it gets to the autopsy. Here's one. Each of these holes, cysticercus larvae. Shouldn't be there. They shouldn't be there. So what sorts of things? What are the pathways to get to essentially death with this disease? Oftentimes you'll start seeing seizures, or maybe headaches first. Maybe you'll start complaining of some headaches. Doctor doesn't know what's wrong. You go see specialists. Maybe it's stress. Maybe it's other things. And then you have a seizure. Now, if you don't have a history of epilepsy in your family, this should be a red flag. All right, if you do have history of, if your family has some epilepsy in it, it might be, you might not get diagnosed right away. But seizures, all right? Then you could have cerebral nerve palsy. So now you have necrosis, blocking of nerve transmissions, leading to, muscles and appendages not working like they should, all right? Being erratic. You can have stroke, all right? You can have paralysis. So not just, limbs acting erratically or not having full control out of them, but then paralysis, you can't move, and eventually death. Oftentimes it kind of progresses, headaches, seizures, and then coma. All right, and then at that, maybe, once they get to that stage, they figure out it's neurocysticercosis, and there's usually little that they can do at that point. All right, so treatment is possible if you catch it in the early stage, except in two cases. One, if it gets to the eye. All right, can't really go in and dig them out of the eye. All right. Second, if they get into the brain ventricles. If they go to the brain ventricles, we really can't go in there to remove them. And you can't really treat them with drugs and kill them, because then you're going to have huge inflammatory response that would likely kill you. This is pretty significant. It's significant in areas where pigs and humans overlap. Backyard poultry farms, normally it's poor areas. However, it just happened in New York. Early 20th centuries, early 1900s, right? Early 20th centuries, you have cases of this popping up in Jewish populations. Why is that kind of odd? Because they don't eat pork. So it's like, why is this happening? Because again, we knew this. We knew Taenia. People knew about the parasites. They knew it went into pork at that time. How did they get it?
The fly eggs taking their food, or the flies taking the eggs? To
No, that would be the nice way. Was it what? No, but a Canadian grad student wasn't happy with their roommate, so they did lace the peanut butter with parasite eggs. So no, it didn't. It was. I'm sure hate crimes were happening, but yeah, this was not a hate crime.
I don't know if they were smart enough to be like, oh,
This was. I should clarify that in the Jewish population, it was hitting the upper echelon of the Jewish population. So now, what do you think? I would say, I think upper middle class would be at like the low end of who we're talking about. What do the rich normally have? The filthy rich. They clean their house. They prepare their own food.
Oh, me.
Yeah, they're housekeepers. They're maids. They're servants. So they would have, again, the working class doing this, and they were paying them. Well, the cooks and the preparers and everything, they were keeping rooftop farms, rooftop poultry, rooftop pigs. Pigs are infected. They're shedding eggs, so the pigs can be a definitive host as well. They're shedding the eggs. So now, how are they transferring it to their, what do you say, their employer? Not washing their hands. There's a book on it. New Guinea tapeworm, Jewish grandmothers and New Guinea tapeworms. I got that backwards. I have that backwards. But it lays it out. New Guinea New Guinea tapeworms and Jewish grandmothers. Yep, that's kind of documents what's going on. Robert Desowitz, he has several books on parasites in humans. Talks about how we kind of figured out what's happening and so forth. So interesting reads. So yeah, wash your hands if you handle pigs. Any questions? And there's a lot. I would invite you to look up cysticercosis or neurocysticercosis. There's a lot of scans and images of it. This is just autopsy section of a brain, but you can also look at X-ray scans, MRI scans. You've got a lot. All right, so our next tapeworm, also Taenia, is Echinococcus. All right, this is the smallest tapeworm, actually. We have it. It consists of basically 3 proglottids. You've got your scolex in your neck, and then you've got immature proglottid, a mature proglottid, and a gravid proglottid. Very simple. Immature is 1, mature is 2, gravid is 3. All right, we do have this. Our specimen's really overstained, but one of the things you can do is go to that gravid proglottid and zoom in, and you can actually see the eggs. Same thing with our Taenia sections. So what we'll do is pick this up on Monday. We're going to zoom ahead. Hymenolopus, we're not going to talk about. So I think I put it on the notes that since we had Hymenolopus nana presented in lab, we're going to leave that for lab, and we're not going to go over like life cycle or anything. And then we'll have our Diphyllobothrium, and I'm hoping to finish all this up on Monday. But if not, we'll go into Wednesday.
This is the last PowerPoint that's gonna be on set, right?
What's that? Yep. This will be it. So you can kind of check it out. Again, Echinococcus, Diphyllobothrium, those are fairly simple life cycles. So, all right. I think is there a bio lunch today? I think there is. There's a bio lunch today on skull morphological changes in captively reared mammals. I don't know if that's the exact title, but that's the focus. Morphological measurements of skulls between wild-caught animals and those that were bred and reared in like a zoo setting. If you could join. Oh, crap, hold on. Your quiz. Bye. There it is. Let's see. Yours, Hector.
I found the email I sent you. I'm making my drafts. I know it's too late now. I just want you to know it was not.
Okay, well, if you want to go down, if you have time right now, you can take it right outside the office.
I have a record of any honors. I'm sorry.
Okay.
Thank you.
How are you, Dylan? Not too bad.
I like that shirt. I like the color.
Salmon?
I don't know the names of colors, but.
Grapefruit? I'd say grapefruit. All right. Move you guys off. All right. Yeah, they weren't. Just look over it. I'm going to put the key outside my office so you can kind of see what I was checking off. And that's going to be how I basically grade for the exams.